Every crewed space mission carries billions of microorganisms into orbit. They are transported unintentionally by astronauts, scientific equipment, cargo, food, and even the air inside spacecraft. Despite extensive sterilization procedures, eliminating all microbial life is impossible because the human body itself hosts trillions of bacteria, fungi, and viruses that make up the natural microbiome. Since the International Space Station became permanently inhabited in November 2000, NASA and its international partners have continuously monitored microbial populations inside the station. Samples are regularly collected from handrails, workstations, sleeping quarters, laboratory equipment, air filters, and water recovery systems. Scientists then identify the microorganisms present and compare their genomes with identical strains found on Earth to determine whether long-term exposure to space changes their biology. Hundreds of bacterial and fungal species have been identified aboard the ISS. Most belong to familiar groups such as Staphylococcus, Bacillus, Enterococcus, Pseudomonas, and Enterobacter. These microorganisms are common inhabitants of human skin, the respiratory tract, and the digestive system. Under normal circumstances, they are harmless, although some species can become opportunistic pathogens if the immune system is weakened. The ISS presents microorganisms with an environment unlike any naturally found on Earth. The most obvious difference is microgravity. Although astronauts often describe the station as having zero gravity, gravity has not disappeared. At an altitude of roughly 400 kilometers, Earth's gravitational pull is still about 90% as strong as it is at sea level. Weightlessness exists because the station is constantly falling around Earth while traveling at approximately 28,000 kilometers per hour. Everything inside remains in continuous free fall, creating the sensation of floating. For bacteria, this changes fundamental physical processes. On Earth, gravity causes cells to settle, while convection continuously circulates liquids, bringing nutrients toward microorganisms and carrying metabolic waste away. In microgravity, convection is greatly reduced, leaving diffusion as the primary mechanism for molecular transport. This seemingly small difference dramatically alters the microscopic environment surrounding every bacterial cell. Nutrients reach cells more slowly, waste products accumulate differently, and neighboring bacteria experience new patterns of interaction. These altered conditions trigger physiological responses that are rarely observed under Earth's gravity. Many news articles describe these changes as space mutations, but this is often inaccurate. Scientists distinguish between changes in gene expression and actual genetic mutations. Gene expression refers to which genes are active at any given moment. A bacterium may activate hundreds of genes that remain inactive under normal conditions, allowing it to respond rapidly to environmental stress without changing its DNA sequence. A mutation, by contrast, is a permanent alteration of the genetic code itself. Mutations can occur when DNA is copied during cell division or when radiation damages the genome. Most mutations have no measurable effect. Many reduce the organism's fitness, and only a small fraction provide an advantage under specific environmental conditions. Radiation is one of the defining characteristics of spaceflight. Earth's atmosphere and magnetic field block a large proportion of high-energy particles originating from the sun and deep space. Although the ISS still benefits from partial protection provided by Earth's magnetosphere, radiation levels aboard the station remain significantly higher than those experienced on the planet's surface. Ionizing radiation has enough energy to damage DNA directly by breaking chemical bonds or indirectly by generating reactive molecules that attack cellular structures. If the damage is not repaired before the next cell division, permanent mutations may occur. However, bacteria are exceptionally resilient organisms. Many species possess sophisticated DNA repair mechanisms capable of detecting damaged DNA, removing defective sections, and rebuilding them using intact genetic information. These repair systems evolved over billions of years and successfully correct much of the damage caused by radiation before it becomes permanent. This raises an important question. Do bacteria actually mutate faster in space? The answer is less dramatic than many headlines suggest. Studies have produced mixed results. Some experiments report a modest increase in mutation frequency under spaceflight conditions, while others detect little or no significant difference compared with identical bacterial cultures grown on Earth. Current evidence suggests that the space environment does not universally accelerate mutation rates across all microorganisms. Instead, researchers believe natural selection plays a much larger role. Mutations occur continuously in every bacterial population, regardless of whether those bacteria live in soil, oceans, hospitals, or spacecraft. The difference is that microgravity creates new environmental pressures. If a particular mutation helps bacteria survive under these conditions, those cells are more likely to reproduce successfully. Over time, the advantageous trait becomes more common within the population. This process is identical to evolution occurring anywhere else on Earth, except the selective pressures are unique to the space environment. One of the first studies to demonstrate this involved Salmonella enterica. In 2006, NASA launched cultures of the bacterium aboard Space Shuttle Atlantis during mission STS-115. After returning to Earth, researchers compared the space-grown bacteria with genetically identical cultures maintained in ground-based laboratories. The results attracted worldwide attention. More than 160 genes displayed altered activity after exposure to spaceflight. Many were associated with metabolism, stress responses, membrane transport, and mechanisms involved in infection. Researchers then tested both bacterial populations in laboratory mice and found that animals infected with the space-grown Salmonella developed more severe disease than those exposed to the Earth-grown cultures. Media outlets quickly described the findings as evidence that space had created dangerous superbacteria. The study itself, however, made no such claim. The researchers concluded that microgravity had altered bacterial physiology and gene expression, influencing how the bacteria interacted with their host. They did not conclude that Salmonella had permanently evolved into a more dangerous organism. Follow-up studies reinforced this conclusion. Different bacterial species respond differently to spaceflight. Some exhibit substantial changes in gene activity, while others show only modest effects or none at all. Factors such as nutrient availability, temperature, oxygen concentration, and mission duration can all influence the outcome, making it impossible to generalize the results from a single experiment to every microorganism. Nevertheless, the Salmonella experiments fundamentally changed space microbiology. They demonstrated that the physical environment of space can directly influence microbial behavior, prompting NASA, ESA, and JAXA to begin a new generation of experiments investigating how bacteria adapt during long-term missions aboard the International Space Station. One of the most important discoveries made aboard the International Space Station involves Enterobacter buchandensis. This bacterium was first identified in 2016 during an outbreak in a neonatal hospital in Tanzania, where it caused bloodstream infections in newborn infants. Like several other members of the Enterobacter genus, it is considered an opportunistic pathogen, meaning it usually poses little risk to healthy individuals but can cause serious infections in people with weakened immune systems. Between 2015 and 2016, NASA researchers isolated several strains of Enterobacter buchandensis from different ISS modules and performed whole genome sequencing to compare them with strains collected on Earth. The analysis revealed numerous genetic differences, including changes in genes involved in metabolism, nutrient transport, stress responses, and cellular regulation. The findings initially attracted considerable media attention, with some headlines claiming that dangerous bacteria had evolved in space. The scientific conclusions were much more conservative. Although the ISS strains showed clear genetic adaptations, researchers found no evidence that they had become more virulent or posed a greater threat to astronauts than comparable strains on Earth. Most of the observed changes appeared to improve survival under the unique environmental conditions of the station rather than increase their ability to cause disease. Another consistent observation concerns biofilms. Instead of living as isolated cells, many bacteria produce a protective matrix made of sugars, proteins, and extracellular DNA that allows entire communities to attach firmly to surfaces. These structures are known as biofilms. They are extremely common on Earth, forming on rocks and rivers, inside water pipes, on ship hulls, medical implants, and even human teeth. Microgravity appears to promote the formation of thicker and more complex biofilms in several bacterial species. Without normal convection currents disturbing developing colonies, bacteria remain attached to surfaces more easily and continue producing the extracellular material that strengthens the community. This creates significant engineering challenges aboard spacecraft. Biofilms can develop inside water recycling systems, cooling loops, air filtration equipment, and narrow pipelines. Over time, they may reduce water flow, interfere with sensors, accelerate material corrosion, and increase maintenance requirements. Unlike free-floating bacteria, cells embedded within biofilms are also more resistant to disinfectants because the surrounding matrix acts as a physical barrier that slows the penetration of antimicrobial chemicals. For this reason, microbial monitoring aboard the ISS is not limited to protecting astronaut health. It is equally important for maintaining the reliability of life support systems that keep the station operational. Antibiotic resistance is another area receiving significant attention. Antibiotic resistance occurs when bacteria acquire genetic mechanisms that allow them to survive drugs designed to kill them. It is already one of the world's most serious public health concerns, responsible for hundreds of thousands of deaths each year. Researchers wanted to determine whether spaceflight accelerates the development of resistance. Several bacterial species isolated aboard the ISS carry genes associated with antibiotic resistance. However, genomic analyses indicate that many of these genes were already present before launch. Since astronauts naturally transport microorganisms from Earth into orbit, bacteria carrying resistance genes inevitably become part of the station's microbial community. Experiments investigating whether microgravity itself increases antibiotic resistance have produced mixed results. Some studies observed temporary increases in antibiotic tolerance while bacteria remained under simulated microgravity conditions, whereas others detected little or no lasting difference once the organisms returned to Earth's gravity. Current evidence does not support the idea that space routinely produces highly antibiotic-resistant superbugs. Instead, the environment appears capable of temporarily altering bacterial physiology, with effects that vary depending on the species and experimental conditions. Viruses present a different challenge. Unlike bacteria, viruses cannot reproduce independently and require living host cells to complete their life cycle. This makes controlled experiments considerably more difficult. One of the most important discoveries came from studying astronauts rather than viruses themselves. Medical monitoring has shown that during long-duration missions, several dormant viruses can reactivate inside the human body. These include Epstein-Barr virus, varicella-zoster virus, and herpes simplex virus, all members of the herpes virus family. Most people become infected with these viruses during childhood. After the initial infection, they remain dormant inside nerve cells or immune cells for decades, usually without causing symptoms. Blood and saliva samples collected before, during, and after space missions revealed that astronauts sometimes begin shedding these viruses while living aboard the ISS. Importantly, this is not caused by viral mutation. Researchers believe the primary reason is temporary suppression of the immune system. Long-duration spaceflight exposes astronauts to multiple physiological stressors, including microgravity, elevated radiation, disrupted sleep cycles, confinement, altered nutrition, and psychological stress. Together, these factors can reduce the immune system's ability to keep dormant viruses under control, allowing them to reactivate. Fortunately, these reactivations have generally produced mild or no symptoms, but they demonstrate that spaceflight influences not only microorganisms but also the human body's ability to respond to them. After more than 2 decades of continuous research, the overall scientific picture has become much clearer. Microorganisms unquestionably change in space. Their patterns of gene expression shift, some acquire genetic adaptations, biofilm formation often increases, and their behavior can differ substantially from identical organisms growing on Earth. These effects are real and have been confirmed by numerous experiments conducted by NASA and other space agencies. However, there is currently no credible scientific evidence that space routinely transforms ordinary bacteria into dangerous superbugs capable of threatening humanity. The changes observed so far are best explained as normal evolutionary adaptation to an unusual environment rather than the creation of entirely new pathogens. The International Space Station has therefore become one of the most valuable microbiology laboratories ever built. By studying how microorganisms respond to microgravity, radiation, and long-term isolation. Scientists gain insights that are impossible to obtain under Earth's gravity. This research not only improves the safety of future space missions, but also contributes to our understanding of bacterial evolution, biofilm formation, antibiotic resistance, and infectious disease here on Earth. The reality is less sensational than many headlines suggest, but scientifically far more interesting. Space does not create monsters. It provides a unique environment that reveals how remarkably adaptable microbial life can be when confronted with conditions unlike anything found on our planet.